to today's lab. Today we'll be talking about laboratory safety in microbiology lab. So let's begin. And before we continue, please like, comment. You can also subscribe and share. Safety in a microbiology laboratory is important in the prevention of infection that may be caused by the microorganisms being studied. Remember, in microbiology lab, we are talking about microorganisms, and these are pathogenic, therefore they can be harmful, especially when we are exposed. So that is why it is usually advisable to avoid using of highly virulent human pathogens. Use the basic ones, the ones that can be handled. And therefore, that's why in the most school laboratory, microbiology laboratories, it is usually advisable to use some of the basic and normal flora bacteria. Therefore, we also have general laboratory safety practices, and we have a lot here, but we'll just talk about them, a few of them. There are those basic ones that we know that we're not supposed to eat, drink, smoke, or chew pens in the laboratory. And therefore, before we continue, let's take note that if you are taking immune suppressants, you could be pregnant, you could be having a medical condition that can compromise your immunity or will prevent full participation in the lab, you need to contact the course instructor before the first day of the lab. Now, before you come to class or to the lab, it is usually advisable to read and understand each laboratory exercise so that you'll be equipped with what to expect. It is usually advisable to use closed shoes, and that is because here we'll be using chemicals, and they could spill and maybe could get inside your shoes. And therefore, it is usually advisable to wear closed shoes. The hair should be tied backwards, those with long hair, and for gentlemen, it's not advisable to wear hats while attending laboratories. We also have personal electronic devices will be turned off and stored while in this laboratory. Therefore, you should not be using your phones, tablets, laptop in the lab. We should also be able to know the specific points or locations of safety equipment. This is because of any accidents we could be having maybe conditions in the lab that require these safety equipments. Therefore, it's usually advisable to have and know the location of these equipments. These equipments that are usually a must that should be found in the laboratories are fire extinguishers, safety showers, and eye wash station. Alright, so we should also recognize the international symbols of biohazards and know where and how to dispose all of the waste materials, particularly the biohazard. They should not come into contact with other wastes. Therefore, they should be separated because the mode of disposing biohazard wastes is usually different from the others. Alright, so I think those are the basic ones. I want us to talk about now the specific microbiology laboratory safety practices. And here I want us to go one by one so that we can understand. So what should be in the back of your head is that all bacteria are potential pathogens that may cause harm under unexpected or unusual circumstances. If you as a student have a compromised immune system or a recent extended illness, you should share those personal circumstances with your lab instructor. because you may be exposed and then you may fall sick, and your immune could be compromised. Number two, you should wear gloves while working with cultures, and when your work is completed, dispose of the gloves in the biohazard garbage. And you should dispose gloves after wearing them. And these gloves should not be worn for a long period of time, and they should not be shared. So you should also have to know how to wear and how to remove these gloves before and after use. It is usually advisable to also have lab coats. Safety glasses or goggles are also required. Now these ones will prevent you from being exposed from the spills or from the bacteria. You should disinfect your work area both before and after working with bacterial cultures. Now in some lab sessions you could be using maybe live microorganisms, and all those live cultures and those materials that we use to handle live cultures must be properly sterilized after use in the laboratory. Here we want to avoid transferring bacteria to other places. Your instructor will always inform you of these specific procedures, and therefore you should follow the following general rules in the microbiology lab. Glassware such as test tubes, bottles, and flasks may be reused and washed after sterilization. In the next lab, we'll be talking about how to sterilize. Therefore, we'll talk about that next class. Now, these are normally placed on a cart at the front of the laboratory after you finished an experiment or an exercise. Be sure to remove labels before placing any glassware on the cart, so that later they can be sterilized and then this glassware could be washed. 2. Some materials such as plastic petri dishes, plastic pipettes, microscope slides, and swabs are considered disposable. These are used once, and if they become contaminated by being in contact with live microorganisms, they should be sterilized and discarded. All of these disposable contaminated materials should be placed in the designated waste containers containing a biohazard autoclave bag. Now it is not guaranteed that all laboratories will be having this autoclave bag, but still it is usually advisable that before you dispose any biohazard, it is usually advisable to sterilize them so that we minimize the exposure to other people. Now, you are not allowed to place contaminated pipettes or pipette tips, inoculating loop or needle, or other contaminated material on the bench top. Sterilize these loops before and after use, and we sterilize these ones using the bactinerator, that is use of high temperatures. You should place the contaminated pipette tips in the orange biohazard buckets on your bench. That is if your laboratory can manage. You should place all other contaminated materials in their designated waste containers. Do not place or put anything containing live microorganism in the sink, because they could grow. Maybe they could be washed down into the sewage, they continue growing, and then they'll be recycled back. So some laboratories could be having aerosols, therefore they should be avoided by use of a proper technique for sterilizing the inoculating loops, and by performing any mixture of cultures and reagents in such a way as to avoid splashing. So remember, we inoculate loops or inoculating loops. We sterilize inoculating loops using bacterincinerators. That's for around 5 seconds, and then allow it to cool before continuing. The cultures or reagents should always be transferred with an automatic pipette that will be provided. We should never try mouth pipetting. You could inhale or take in bacteria. The bacteria should always be kept, the cultures, they should always be kept in proper storage racks when not being used during an exercise. Now, sometimes we could be having accidental spills involving a bacterial culture. It is usually advisable to saturate the spill area with disinfectant and then cover the area with paper towels and allow the spill to sit for 10 minutes. Later, you should carefully remove the saturated paper towels and dispose them in the biohazard waste. And then the area should be clean again with the same disinfectant or another one. But it is usually not advisable to use two disinfectants because they could neutralize one another. And then let the instructor know about the spill. If the chemical is marked danger or caustic, you should notify the instructor who will handle this type of spill. You should immediately report all accidents such as spills, cuts, burns, and other injuries to the instructor. Make sure that lab benches are completely cleared. That is after use, after you've done the experiment, and it is also advisable to store the remaining equipments or apparatus in the designated areas before you leave the lab. Now, the type of clothes that we wear in the microbiology lab is very important and should be washed before being subsequently worn in a facility such as hospital, clinic, or nursing home, or in an area of public food preparation. Here it's because in hospitals or nursing, we have people of compromised immunity, those the young ones, and also the elders. Therefore, it is usually advisable to keep those laboratory lab coats. It is usually advisable to keep them separate from the clothes that you usually wear while attending other places or food preparation areas. Now, in the event of a fire alarm, you should follow the directions of your instructor and meet at the place designated by your instructor. You should know the exits, and you should also avoid running in the lab. This could cause accidents, and you could get injured. Those are the specific laboratory microbiology laboratory rules and guidelines. Therefore, it should be followed to ensure a successful exercise in the lab. Thank you very much. My name is John Bosco Chieng. I'm a lab instructor for microbiology lab in one of the universities.